monitoring. Therapeutic drug monitoring is a subunit of second topic. In short, it is called as TDM. All of you visible my screen? All of you heard about the therapeutic drug monitoring in pharmacology subject. It is nothing but it's a ratio of therapeutic effect of drug that can be monitored in individual to individuals. Therapeutic drug monitoring in the syllabus, the content of therapeutic drug monitoring, that is TDM. The content of TDM, that is therapeutic drug monitoring, as per your syllabus is concerned, definitions, what are the needs for the therapeutic drug monitoring, or what are the indications why it is necessary monitoring of the particular drug for their therapeutic actions, and what is factor to be considered during the therapeutic drug monitoring, and current India scenario for therapeutic drug monitoring. These are the contents. These are the content of your syllabuses. After finishing of this TDM, you have to learn about the therapeutic drug, what is therapeutic drug monitoring and their need for the management of TDM for concerned patients of therapeutic effect of concerned drugs. Another things you have to learn from this topic, that various factor to be considered during the therapeutic drug monitoring and current Indian scenario for the therapeutic drug monitoring. These things you have to learn after finishing of these sessions. So let's start with the therapeutic drug monitoring. What is meant by therapeutic drug monitoring? Therapeutic drug monitoring, nothing but is a measurement of the plasma concentration. Which concentration? Drugs. Plasma concentration level of a drugs and the coordination of the serum level with the serum therapeutic range. Means whatever the drugs enter inside our body, they are collected from the blood samples as well as blood tissue and monitor that. And to monitor the effect or drug concentration for their therapeutic effect. That is called as therapeutic drug monitoring. Name itself indicates that therapeutic means effect of concerned drug we have to monitor at plasma concentrations of drugs as well as coordination of the serum level with the therapeutic range of the particular drugs. Why therapeutic drug monitoring is essential? Because TDM can guide the clinician to provide effective and safe drug therapy to the individual patients using their serum drug concentration. That's why this therapeutic drug monitoring is essential for providing the effective and safe drug to the particular individual patients. Why should drug level be monitored? Why? Because in excess plasma concentrations, there is a upper limit of the particular therapeutic window should be toxic one. Means particular drug should be toxic. That's why this monitoring is essential. If lower limit achieves the concentration below the lower limit of the therapeutic range, that particular drug is ineffective to the concerned patients. That's why this drug level monitoring, that is therapeutic drug monitoring, is required. If upper limit, above the upper limit, particular drug concentration reaches, means it indicates that particular drug is toxic to the particular patients. And if the lower limit of the particular concentration of drug achieves lower than the limit, then particular drug is ineffective to the concerned patients. So this type of responses at similar doses we have to monitor in the form of therapeutic drug monitoring. Whenever you monitor the therapeutic drug, therapeutic effect of that particular drugs, there is some range. That range is called as therapeutic range, where the particular effect is safe and effective to the particular patients of concerned drugs. That range is called as therapeutic range, or another word is therapeutic window. The definition of therapeutic window or therapeutic range is the concentration range of a drug plasma where the drug has been shown to the effectiveness without any toxic effect to the particular patients. These two lines in between these two lines, this range is called as therapeutic range. Means effective with lesser toxicity or without toxicity. What are the range are called as therapeutic range. Above this limit, that is the toxic range in therapeutic window, and below the range, below the lower range, a particular dose of the particular drug is a subtherapeutic range, means ineffective of particular drugs. So what are the range in between effective without any toxic effect? That range is called as therapeutic range or therapeutic window. The range in between. Below the range, that is called as ineffective of particular drugs. Above the range, it produces the more toxicity along with some effect. So whatever the therapeutic range, it is nothing but it's called as effective concentration of the particular drugs without any toxic effects. This figure indicates that the serum concentration of particular drugs in between 50 to 25, that range is called as steady state effective range that for the particular drug, that is called as therapeutic range or called as therapeutic window. Another example, another figure related with the therapeutic window. Here we have to see this point to this point. This is the duration of action of concerned drugs. And this part of therapeutic window is a peak effect of concerned drugs within the particular therapeutic window. Means here onset of action of particular drug was started, and here the particular action of particular drugs are lowers. And in between this onset of action started and ended, that is called as duration of action of concerned drugs. This is carried out only in therapeutic window of particular drugs. Above the therapeutic window, above the limit of the therapeutic range, that is called as toxic effect of concerned drugs. So this matter is considered during monitoring of particular drugs of their therapeutic effect. That is therapeutic range of concerned drugs. This therapeutic range, where we have to find out, this range is required for the monitoring. Generally, this therapeutic range recommended are generally found in the product inserts for drug that require monitoring. Whatever the products available for the monitoring, they give the insert of particular products range. From that, you have to identify the information regarding therapeutic range of concerned drugs, or certain books, official books should be available, some primary medical journals, some reference journals, some articles. From that, you have to get the information related to the therapeutic range of particular drug. Why it is necessary? TDM, therapeutic drug monitoring. This is a necessary need of the TDM, therapeutic drug monitoring. So, are various indications where TDM is required. Means first possibility or first need, the drug for which A relationship between dose and plasma concentration is unpredictable. In that conditions, TDM is requirement. For example, phenytoins, there is a dose and plasma concentrations are unpredictable to their therapeutic effect. In that condition, TDM is necessary. Another conditions or another need for the TDM, the drug with a narrow therapeutic windows. In that conditions, we have to monitor drugs by altering the dosage forms to give the optimal therapeutic effect by avoiding the toxic effect of particular drug. For example, digoxins, phenytoins, lithium. These are drugs having the narrow therapeutic index. For achieving this narrow therapeutic window drugs, we have to change or alteration in the dosage forms and to avoid the to give the optimal therapeutic effect with lower toxic or avoiding the toxic effect of particular drugs. That type of narrow therapeutic window we have to needed the TDMs. Another need of TDMs or indications, drug with sleep dose response curve. For example, theophyllis. Theophyllis produces the sleepness while during the administration of drugs. So in that conditions, to avoid the sleep response of particular drugs, you have to monitor the TDMs and adjust the dose according to the therapeutic effect without their unwanted effects. Another conditions where you have to monitor the TDMs, therapeutic drug monitoring, drug for which there is a difficulty in measuring or interpreting the clinical evidence of therapeutic or toxic effects. For example, nausea and vomiting occurs in both digitalis, toxicity, and congestive heart failures. In that conditions, we are too difficult to measure the toxic effect or therapeutic effect. So in that conditions, TDM is requirement. For example, digitalis, toxicity of digitalis produces nausea and vomitings as well as congestive heart failures. In that conditions, this TDM is necessary or needed. Another conditions where TDM is needed, drug with saturable metabolisms. Example is phenotypes. Another conditions, drug with poorly defined endpoints or difficult to clinical predict the response. In that conditions, TDM is needed. For example, immunosuppressant drugs gives the less defined endpoint related to the response. In that conditions, TDM is necessary. Another condition is renal diseases. In renal diseases, TDM is requirement in the alteration of the dose along with their plasma concentrations. For example, in renal diseases, digoxin, lithium, aminoglycoside antibiotics, TDM is requirement or needed. Another need of TDM is during the drug interactions. When particular drugs alter the action of other drug and then concentration in the plasma. In that conditions, this TDM is required. That is drug interactions. In that example is plasma concentration of lithium is increased by thiazide diuretics. So in that conditions, in the drug-drug interactions, TDM is needed. This TDM is individual to individual, varies. Patient to patient varies. The monitoring effect. Drug with large individual variability at steady state means the therapeutic range, that is plasma drug concentrations in given dose. Means availability of the large dose is individual to individual varies. Another needed for the TDM for diagnosis of substituent toxicity and determine drug abuse conditions, TDM is necessary or required. Another is to evaluate the compliance of patient. Means particular patients comply with the particular drugs. Means so and so drugs is harmful to me while taking the particular medicaments. In that conditions, evaluate the TDMs, identify the dose, at what dose, particular harmful effect to the particular patients. You have to identify, reduce the quantity of the particular dose, ultimately reduce the concentration of the plasma of particular dose and identify the monitoring dose. And finally, in withdrawal of the therapy, particular therapy is withdraw, suddenly withdraw. In that conditions, TDM is needed. For example, anti-epileptic drugs, cyclosporines, these are the drugs are immediately withdrawal of therapy. In that case, TDM is required. Means as per your syllabus is concerned, these two slides it's need of TDMs, where these are the 10 to 12 points required for the requirements or needed of the TDMs at various indications. Yes, there is alterations, drug interactions, heavy dose, unpredictable response. In that conditions, generally TDM was performed by guiding therapeutic range. आता आपण बघितलेलं आहे TDM is required. Where? TDM is not suitable for particular drugs. Means particular drugs should not be suitable for TDMs, therapeutic drug monitoring. Give exceptionals in TDMs monitoring. Means drugs that are used for the treatment disease of which their clinical endpoints. Means for a sugar patient, diabetic patient, they give the blood report of diabetes, sugar levels. In that conditions, there is no need of the TDMs. Like blood pressure, higher blood pressures, they give the particular medicaments related to higher high BP controls. That drugs are given. In that cases, the TDM is not suitable, not advised. Pain, temperature, headaches, urine volume. In that case, TDM is not suitable. Or these are the drugs which are used for the clinical endpoints. In that cases, TDM is not used or not suitable. Another conditions where TDM is not required or not suitable, that is drug whose serum concentration does not correlate with the therapeutic effect or toxic effect. Means whatever the drugs given are available in the serum level, serum concentration, that drug does not affect their therapeutic effect also as well as toxic effect, does not produce. In that conditions, TDM is not suitable. Another condition, drug with less complicated pharmacokinetics. In that conditions, there is no need of TDMs. Drug having wide therapeutic index means particular drug having the different axes. In that case, TDM is not suitable. And hit and run drugs, for example, omeprazoles, they hit and immediately run to the. In that case, the TDM is not suitable. So where the TDM is required, where the TDM is not required. These are the not requirement of the therapeutic drug monitoring. जिथं मॉनिटर होतं आहे क्लिनिकल एंड पॉइंट आयडेंटिफाय होते हैं तिथं लागत नाही आपल्याला TDM. Next point, our next consideration is basic principles of TDMs, therapeutic drug monitoring. There are 4 principles where the therapeutic drug monitoring was carried out. First principle is measurement of patient serums or plasma drug concentration taken at an appropriate time. after drug administration. Means whatever for the measurement of the particular drug monitoring, the measurement of the plasma drug concentration is essential, where the appropriate times the plasma concentration was taken by the particular laboratory persons after giving the dose or after drug administrations. At particular times, the particular plasma concentration of particular drug you have to collect it. Another principles behind the therapeutic drug monitoring is knowledge about the pharmacological actions as well as pharmacokinetic profile of the administered drugs. Means whatever the drugs are administered, their pharmacological and pharmacokinetic activity or profiles should be known by the clinicians. Knowledge of third principles, knowledge of relevant patients profile like demographic data, clinical status, laboratory and other clinical investigations should be know about that particular patients. And interpretation of SDC, that is serum drug concentrations. These are the four principles you have to use during the therapeutic drug monitorings. And this information is individual to individuals are vary, individualizing drug regimens according to the clinical need of the particular patients. This TDM was carried out. What is the significance, clinical significance behind the TDMs? Major effect is maximum efficacy of the particular drugs. Means why this TDM was carried out? Because it gives the maximized efficacy with lower dosage. Second, ultimately, second is efficacy only without toxicity, avoiding the toxicity of particular drugs. Third importance or significance of TDM is identifies therapeutic failures. Whatever the non-compliance, subtherapeutic doses, we have to identify their therapeutic failures. Facilitate adjustment of the dosage. Means whatever the therapeutic effect you have to adjust as per the plasma concentrations. Means new dose is equal to old dose into desirable steady state, clinical steady states, divided by old CSS. This formula is utilized for the adjustment of dose. Next significance is facilitate the therapeutic effect of drug by achieving the target drug concentrations. And finally, identify the poisonings, drug toxicity, and drug abuse. These are the clinical significance of therapeutic drug monitoring. Measure is maximize the efficacy, minimizing the toxicity, identify the therapeutic failures, facilitate the adjustment of the dose after TDMs, identify the poisoning, drug toxicity, and drug abuse, and facilitate the therapeutic effect of particular drugs. These are the process of TDMs. What is the process we have to carry out for TDMs? For development of plasma profile in each patient. First is administering a predetermined dose of the drug. First, give the dose to the particular patients whose undergo plasma profiles for TDMs. Firstly, administer the predetermined dose. After administration, collection of the particular blood samples at particular times after administration of the drugs. Then determination of the blood samples and plasma profile and pharmacokinetic model developments. that is, which shows clinical effect of particular drugs, development or alteration of the dosage regimen to the particular drugs, and diagnosis, dosage form selections, dosage regimens, and initiation of the therapy. This was carried out in TDM to evaluate the clinical response to the particular patient after processing with the plasma profiles to evaluate the clinical response of the particular patient to the particular drugs. These are the process of TDM. These are the drugs which requires the TDMs. Some examples. Digoxins, their therapeutic range is 0.5 to 2 nanogram per ml. Lidocaines, 1.5 to 5 micrograms per ml. Means these are the drugs having the different effect to individuals. Some gives unwanted effects, some gives better effects, some gives does not affect on the particular body. That that should be monitored. Require the TDMs. These are the examples. Anyone can take the screenshot of this examples of TDMs along with their range, therapeutic range, where the particular concentration gives therapeutic effect without toxic effect of that particular drugs. These are the management of therapy using plasma concentrations on the basis of the clinical response. Request to the laboratory for analyzing the plasma samples, plasma levels. In analysis, quality assessment is required. Then after analyzing, report to the particular plasma profiles, their interpretation with the help of clinicians, and do the necessary clinical actions. After taken the clinical actions, once again go for the laboratory plasma collections, analyze, report, and whether this TDM earlier collected, older reports or new reports, you have to interpret that, and again adjustment of the dosage of particular drugs to the individualized patients. This is a management of therapy using plasma concentrations. And these are the request form of TDM. In the request forms, it requires patient names, date, age, along with drug level requested, reason for request, either toxicity or compliance, patient compliance related to particular drugs, therapeutic confirmations, or absence of therapeutic response to the particular patients. For that reasons, this TDM is requested by analyzing the plasma samples. At particular date and times, large dose is given, either in the form of IV, IM, subcutaneous, and sampling time after taking the last dose. At what time particular sample and at what date particular sample is collected for TDMs. These are the forms for request therapeutic drug monitoring. Can drug concentration in other fluids of body can be measured? Yes. Other fluids of body can be measured the drug concentrations. For example, in urine, you have to measure the drug concentration of benzodiazepine drugs. In sweat, you have to identify the drug concentration of cocaine and heroines. In saliva, cocaine's alcohol, marijuana drugs should be identified or measured. In breathing, alcohols, all of you seen in the pictures, there is a alcohol monitoring machines. They are monitor the alcohol concentrations after breathing, long breathings. That type of measurement of the particular drugs you have to carry out their concentrations. And finally, TDM in India. The first monitoring of drug therapy, therapeutic drug monitoring, was introduced in 1980s, the first TDM. And overseen the in last 20 years, they are having the better growth. In India, TDM is mainly two type of settings. First is in large teaching hospitals. Through the Department of Clinical Pharmacology, this therapeutic drug monitoring was carried out in large teaching hospitals. Means those are the MBBS colleges having the Department of Clinical Pharmacology. On that for pharmacology, clinical pharmacology department, this therapeutic drug monitoring was carried out. And thing is in private sectors, in biochemistry. In private sector, biochemistry lab dedicated to central processing unit in corporate hospitals. Corporate hospital like Apollo, examples. In that sector, this TDM process was carried out in India. Majorly in large teaching hospitals generally.
नाही आम्हाला थोडस मोठ्याने बोला ना सर प्लीज, काहीच समजत पण नाही असं हळू आवाजामध्ये.
कस काय? स्पीकर छोटा आहे का तुमचा मोबाईलचा?
म्हणजे आवाज हळू यायलाय.
आवाज ड्यू टू इंटरनेट प्रॉब्लेम असू शकतो तुमच्याकडे किंवा माझ्याकडे. माझा फोटो बघायलात का तुम्ही? व्हिडिओ दिसते का माझा? एकदम माईक माझ्या समोर ठेवलेला मी तोंडासमोरच. स्पेशल हे डिझाईन करून घेतलंय आयडेंटिफाय करून तुमच्यासाठीच विथ माईक असलेला हेडफोन घेतलाय मी, जेणेकरून तुम्हाला प्रॉब्लेम येऊ नये. आणि माझ्या लॅपटॉपचा आवाज पूर्ण आहे, आउटपुट इकडून तरी काही प्रॉब्लेम नाही. ड्यू टू इंटरनेट इंटरप्शन्स हा पॉसिबल येऊ शकतो, पॉसिबिलिटी आवाज. त्याची काही काळजी करू नका. मी परत माझा व्हिडिओ इथं अपलोड करतोच आहे आपल्या गुगल क्लासरूमला, तिथं परत एकदा ऐका तुम्ही. जरी इथं मिस झालं तरी तिथं बघा. आणि काही गुगल क्लासरूमला मी नोट्सही टाकलेले आहेत, नोट्सही काही टाकलेले आहेत वर्ड फाईल मधल्या तुम्हाला वर्कआउट करण्यासाठी, त्याही थोडं बघून घ्या. अलग विथ पीपीटी टाकलेले आहेत. काही जणांचे डिमांड होते सर पीपीटी टाका, त्याही पीपीटी टाकलेले आहेत. आणि काही नोट्सही डिटेल मध्ये जे अवेलेबल झालेत माझ्याकडे, जे मी काढलेले आहेत स्वतःहून, ते मी टाकलेले आहेत. जेवढे अवेलेबल असतील ते डिटेल मध्ये. अदरवाइज पीपीटी टाकलेलेच आहेत. आणि काहीच्या डिटेल मध्ये माझ्याकडे नोट्स बनवायचे आहेत, त्याच्यामुळे टाकलेल्या नसतील असं कन्सिडर करा. ठीक आहे. दिस इज अ पार्ट ऑफ थेरापेटिक ड्रग मॉनिटरिंग इन इंडिया. या टॉपिक मध्ये आपल्याला थेरापेटिक ड्रग मॉनिटरिंग म्हणजे काय, व्हाय इट इज नीडेड, अँड द करंट सिनेरियो इन India. Current scenario in TDM in India was started in the 1980s. In the last 20 years, it's in the higher growth in the TDM monitoring of particular drugs with better efficacy of the particular drugs to the particular patients. So this today's sessions I had to uploaded in Google Classrooms along with the PPT. This is related with the TDMs. Okay. अजून सांगायचे आज फक्त 47च आहेत सध्या पार्टिसिपेंट्स किंवा स्टुडंट्स. गुगल क्लासरूमला फक्त 67-68 जणांनी अपलोड केलेले आहे. की अटॅच झालेले आहेत गुगल क्लासरूमला. तर बाकीचे जे राहिले असतील ते बघून घ्या, कारण नेक्स्ट वीकला तुमची एक्झाम आहे. डेट काय आहे आपल्या एक्झामची? फार्मसी प्रॅक्टिस